We are Group GHR, Olivia and McKenna, and this is our final presentation on our findings regarding our brachipodium gene from our semester of gene and genome analysis.
To begin our research on the unknown gene, we utilized two reverse genetics approaches to better understand the structure of our gene. Reverse genetics is important because structure informs function, and our goal in this experiment is to identify the function of our unknown gene. The first ab initio approach used MBOS FGenesh, and it predicted a sequence including 7 exons and a total of just under 5,000 base pairs as seen in figure 1. The 7 exons were mostly in the second portion of the sequence, and after intron splicing, a 993 base pair sequence remained. To confirm these predictions, homology-based approach was consulted, displayed in figure 3. Phytosome BLAST produced similar results, however, there was some discrepancy in the reading frames as seen in figure 2. Because of this, we ended up sticking with the ab initio approach prediction and used the corresponding sequence in our initial working map. The two approaches both confirmed our unknown GHR gene as Brady1G 68250.1, and this identification directed the rest of our research.
Using Phytosome BLAST, we identified many mutations with varying levels of impact on gene expression. One mutation was selected for further analysis, as you can see highlighted by the red arrow here. This heterozygous mutation was a high-impact donor splice site mutation with a guanine nucleotide in place of a thymine. This mutation would potentially prevent the intron from being spliced out during transcription.
Literature searches on our Brady gene highlighted the lack of consistency in a conserved domain within our gene. Because of this, other sources were consulted to establish gene function predictions. NCBI, EBI, the Swiss model, and Fury2 server all had different outputs on the family that our gene belonged to. However, they all agreed that the gene functioned as a GTPase. The EBI and Fury2 server both produced model predictions of the gene that were strikingly similar as seen in these figures. These sources helped us to identify that our gene was likely a phosphate binding loop GTPase of the TERFAC class, which are important regulators for signal transduction, cell growth, and differentiation.
The DNA from all 6 of our mutant brachipodium plants was extracted and quantified to better understand the gene expression. Mutants 2, 5, and 6 had the highest concentrations of extracted DNA according to the gel electrophoresis shown here. It is important to note that our gel electrophoresis data was compared to a ladder that was run on a separate gel due to the ladders on these gels not running correctly. The nanodrop and fluorometer data also confirmed that the strongest mutant samples were mutants 2 and 6. Therefore, mutants 2, 5, and 6 were selected for further sequencing to confirm the gene functionality.
In order to visualize structural differences between the wild-type and mutant Brachipodium distachyon plants, the middle internodes of all the plants were plucked and cut into cross-sections. These cross-sections were stained with T-blue and PHHCl. We noticed that the mutants have thinner cell walls in comparison to the wild-type plant internodes, as pointed out by these arrows. Our gene is responsible for cell growth, and we believe that our mutation causes the thinner cell walls and the altered shapes of the mutant cells as shown in these microscopy images.
Using the EFP browser, we were able to find that our gene is most highly expressed in the roots and young leaves, as you can see in figure 1 here. The Hazen lab data also confirmed that the expression was highest in the roots. This data is shown in the graph labeled figure 2 here. The p-values that we calculated with Bonferroni correction concluded there were significant differences between the gene expression in the leaf versus root, root versus stem, and stem versus leaf.
Based upon our research thus far, we conducted an experiment to help confirm our gene function predictions. We decided to use an indole-3 acetic acid treatment as it plays a role in cell growth and differentiation. Since our predicted gene function codes for the enzyme P-loop GTPase, which is involved in cell growth and differentiation and is expressed mostly in the young leaves, we hypothesized that the IAA would have an enhancing impact on the young leaves it was sprayed onto. We began by spraying 100 microliters of 10 micromolar IAA solution in ethanol onto the leaves of half of our plants named the treated group. The other half was our control group, which were sprayed with 100 milliliters of water. After 3 hours had passed, we analyzed the wild-type leaf growth of the control and IAA-treated plants.
The young leaves from the control and treated plants were excised and their RNA was extracted and quantified. It is important to note that the tube containing treated 2 sample had little RNA present due to a hole in the test tube during thawing. According to the gel electrophoresis, controls 2 and 3, as well as 1 and 3, had 2 clear bands and significant RNA concentrations, which confirms successful RNA extraction. The nanodrop results confirm that all 3 controls had high RNA concentrations, as did the treated 1 and 3 samples.
In our experiment, we hypothesized that the IAA treatment would create higher gene expression in brachypodium plants. After the Hazen lab ran the qPCR, we analyzed the data and calculated the fold change for our control and treated plants, as well as for wild type and mutant controls. The treated group had the highest fold change as seen in this graph. We conducted t-tests to produce p-values comparing the control to the treated and the wild type to the mutant. It was found that neither p-value had statistical significance, so we rejected our null hypothesis and concluded that the IAA in this treatment did not create higher brachypodium gene expression in the treated plants.
Based upon the experiment results with the IAA treatment, the functionality could not be confirmed due to a rejected hypothesis. Despite this, there is strong evidence that our gene has GTPase functionality involved in the early growth and development of the plants. Hazen lab data, as well as EFP browser results, confirmed high gene expressions in the young leaves and roots. In order to fully confirm the role of our gene as a GTPase within the Brachypodium distichum plant, further experimentation and research would be needed to be conducted. Our research was able to identify the unknown gene as Brady1G68250.1, quantify it in plants, and analyze different aspects of the plant in order to make a stronger prediction on its functionality as a GTPase protein regulating cell growth and differentiation in Brachypodium plants.
This slide pulls together all of the sources we utilized in our research, including the software programs we used to analyze our gene structure and function. Olivia and I would like to thank Dr. Verma, Ahmed, and Kate for their support in this research on our Brachypodium gene.